Welcome back to Science Lullaby. Tonight we take a calm walk through a quiet library written in the language of life, a place where every cell keeps its own book of instructions, and where a new set of tools helps careful readers fix a letter, a word, or a line when nature's printing press makes a typo. This story begins with small creatures and a clever trick, and it grows into a gentle exploration of how we might mend illness, protect harvests, and care for ecosystems, all while asking clear questions about when to proceed and when to pause. Settle your thoughts. Let the mind float, and imagine each breath turning a page. In the oceans, the soil and the air that surrounds us, bacteria face relentless waves of viruses, tiny invaders that try to take over the cell's machinery. Long ago, bacteria learned to keep souvenirs from these battles, little fragments of enemy code tucked away between short repeating patterns, like snapshots in an album. These snapshots are guides, and when danger returns, the bacterial machinery copies a guide and pairs it with a protein that can find and cut matching sequences, removing the threat with surgical focus. This memory album and its search and cut duo is what scientists call CRISPR and Cas, a natural security system that remembers adversaries and responds with precision. For a long time, it was an ecological curiosity, a chapter in microbiology that felt complete and self-contained. Then researchers wondered, what if this search and cut capability could be retuned to find any chosen word in the genome's book? What if the guide could be written by human hands, pointing the protein to a specific page, a specific line, a specific letter? With that shift in perspective, CRISPR became a programmable editor, a penknife for DNA, simple to aim, easy to retool, and powerful in its consequences. Picture a vast encyclopedia that spans from the first cell you ever were to the tissues you carry now. A single misplaced letter can change the meaning of a paragraph, and a small correction can restore the original sense. CRISPR's promise is not the drama of rewriting entire chapters, but the quiet art of fixing a line that went wrong. Scientists learn to craft short guide RNAs, tidy little ribbons of code that tell the protein where to land, where to open the page, where to place the cut. Sometimes the cell seals the cut quickly, like a book whose page is tape repaired, leaving a few letters missing or added, which can switch a faulty instruction off. Other times the cell can be given a little template, like a sticky note with the correct spelling, and as the page is repaired, the right letters are copied in E. This is the heart of editing, either snipping to silence a harmful instruction or pasting in a corrective phrase that restores a lost function. In this way, the lab bench became a repair desk for written life. From the lab came the clinic, first through a careful route. Instead of editing inside the body, doctors took cells out, made the correction in a controlled environment, checked the results with patients, and then returned the cells to the patient. For conditions like sickle cell disease or beta thalassemia, where faulty instructions in blood stem cells bend red blood cells out of shape, ex vivo repair offers a path to relief. Here the analogy is a fleet of tiny boats built in a harbor, strengthened and fixed where wind and waves cannot interfere, and only then sailed back into open water. The early successes of this harbor method reassure us that precise editing can be both effective and safe when every step is measured. Gradually, technologies are moving closer to in vivo approaches, where the repair might happen inside the body itself. And this is where a new challenge becomes the main character in the story. Delivery. Delivery means guiding the editor to the right organ, the right cell type, the right address, and doing so with gentle timing and minimal collateral effects. It is one thing to own a fine set of tools and another to carry them into a narrow attic without scratching the walls. Virus is engineered for carrying molecular cargo, lipid nanoparticles that can ferry instructions like folded letters, and other vehicles are all part of a growing toolkit to reach difficult destinations. The liver and blood are nearer ports of call, more accessible, and already familiar to our delivery systems. Other organs remain distant towns on a winding map, protected by barriers, and vigilant against unknown visitors. Alongside delivery comes the need to tame off-target edits, those unintended cuts that change a word in a paragraph we did not mean to touch. Advances in enzyme engineering, careful guide design, and smart measurement methods keep tightening the circle of accuracy, bringing the cut only where it belongs. Immune responses also ask for attention, because any visitor, even a helpful one, should knock softly before entering. The path forward emphasizes transient presence, tools that arrive, do their work, and leave without lingering, so the book remains stable and untroubled after the repair. Now invite another collaborator into the room, one that does not breathe, yet reads patterns with tireless focus. Artificial intelligence helps us predict where a guide will land, how certain a cut will be, and what side effects might follow. Think of AI as a reading lamp that highlights the page, revealing faint watermarks that the eye alone might miss. Models trained on vast datasets learn sequence features and structural hints that decide whether a target is precise or risky. These systems can score potential guides, rank them for expected accuracy, and even suggest edits to the editor itself, refining the protein so it behaves with greater grace. In design, AI becomes a CEO author, not replacing human judgment, but offering drafts that are more likely to fit the goal on the first try. In analysis, it becomes a proofreader, flagging places where a stray mark may have slipped in and... Together, wet lab craft and computational foresight shorten the journey from concept to clinic, saving time, reducing uncertainty, and focusing attention where it matters most. Beyond medicine, the same logic of gentle revision touches fields that feed and sustain us. Crops that face drought, heat, or pests may benefit from edits that change how a plant uses water, how it resists insects, or how it stores nutrients. Instead of spraying broad solutions across a field, editing lets us adjust the plant's own recipe, strengthening resilience from within. In agriculture, the analogy shifts from repairing a book to tuning an orchestra, nudging a few instruments so the whole performance stays in harmony during a storm. In ecology, editing can help restore balance where invasive species or diseases upset local systems, though here caution grows stronger and the circle of stakeholders widens. Gene drives and other population-level tools are powerful, and they ask for layers of oversight, reversible designs, and community consent before any real-world use. Used thoughtfully, genome editing could help protect biodiversity, support sustainable yields, and reduce chemical inputs, but every step should be measured and transparent. At this point, the mind often turns toward a question that carries special weight: the possibility of changing genes that pass to future generations. This is called germline editing, and for now, it remains beyond the boundary of safe and responsible practice. Early experiments in embryos make clear that the technology does not yet deliver the uniform accuracy or the predictable outcomes required for such profound changes. Even if the technical hurdles were smaller, the ethical and social dimensions remain large. A decision that echoes across descendants is not only a technical action, it is a cultural and moral choice that involves everyone, not just the individuals in a clinic room. The better path today focuses on somatic editing, which affects only the treated person and can be weighed by familiar standards of medical risk and benefit. Meanwhile, transparent global conversations continue, building consensus on governance, drawing lines that reflect caution, equity, and humility. As the tools grow sharper, the guardrails must grow sturdier so that power and wisdom stay in step. To understand why delivery and specificity matters so much, return to the image of the book. Every cell is reading all the time, assembling proteins, guiding development, reacting to signals, and repairing damage. An edit that arrives in the right chapter can restore meaning, but an edit that lands in the wrong place can alter a recipe we did not intend to touch. This is why improved vectors, better tissue targeting, and safer editor variants are the practical frontier. It is also why timing matters, why brief presence can be better than permanent installation, and why dose and distribution are designed like careful brushstrokes on thin paper. Quality control follows every stage, using sequencing to confirm changes, measuring possible off-target sites, and monitoring cells for signs of stress. The workflow is a choreography of checks, where each step listens to the previous one before moving on. As these practices settle, the bottleneck shifts away from whether we can cut and toward whether we can reach. In blood and liver, access is easier, and targets are clearer. In the brain, the heart, the lung, and the kidney, access is harder, and we walk with lighter feet. It is reasonable to expect that delivery technologies will keep improving, inspired in part by lessons from mRNA vaccines and other nucleic acid medicines, where packaging, stability, and targeting have advanced quickly. As more conditions become accessible, we will need basic research to map which genes can be safely adjusted, which pathways tolerate change, and which edits are likely to help more than they harm. The result will be a growing catalog of well-characterized targets, a shelf of tried and trusted repairs that clinicians can select with confidence. In parallel, the community is refining the tools themselves. Editors that nick rather than cut, editors that swap one base for another without opening a full break, and editors that write a small patch using a built-in template offer different levels of subtlety. Each variant trades speed, precision, and byproducts in a different balance, and researchers choose the right instrument the way a surgeon picks a scalpel blade, not for drama, but for fit. Paired with careful guide design and AI-informed scoring, these instruments make it more likely that the correction will be clean, efficient, and limited to its intended site. This is the difference between carving and calligraphy, between bold action and precise strokes that leave the page calm after the ink dries. When you zoom out, the shape of the future becomes clear. We will likely see a steady rise in somatic therapies where the risk-benefit profile is favorable, the targeting is mature, and the monitoring is robust. We will likely see agriculture benefit from targeted resilience traits that support yields with fewer inputs, especially as climate patterns grow less predictable. We may see ecological projects where local communities choose reversible and tightly controlled interventions to restore balance in fragile systems, guided by shared oversight. And we will continue to see thoughtful hesitation at the border of heritable change, a boundary marked by scientific caution and collective ethics. Across all these settings, transparency, access, and fairness will matter, so benefits do not cluster only where resources are greatest. Education and public dialogue will help, because understanding reduces fear, and clarity builds trust. Now let the pace slow a little more. Return to the library image, where each breath is a hand that turns a page without a sound. CRISPR began as a bacterial memory of past storms, and in our hands, it has become a way to mend pages that time or chance have worn. Its power does not ask for spectacle. It asks for care. It allows us to imagine treatments that correct the cause of disease, rather than only softening the symptoms, and crops that endure with fewer burdens on soil and water. It allows us to consider ecological help where species struggle, with community voices guiding the plan. And it obliges us to draw lines, to decide together where the book should never be altered, because inheritance is larger than any one life. As the tools grow finer, and the maps of the genome grow clearer, collaboration with computation will keep brightening the reading lamp, pointing out safe paths through dense paragraphs of code. Delivery will keep improving, editors will keep refining, and safeguards will keep strengthening. With each improvement, the art of repair becomes less disruptive and more like restoration, preserving the feel of the original page even as the meaning is restored. If you are listening in the dark, you can picture a quiet workshop, a bench with a few instruments laid out neatly, each designed for a particular touch-up. You can picture a logbook with careful notes, outcomes measured, uncertainties labeled, and lessons shared. You can picture a circle of chairs, scientists, clinicians, ethicists, and community members speaking in turn, building rules that make the tools safer to use and fairer to access tool. You can picture a field at dawn, crops standing steady after a night of dry wind because a small edit helped them sip water more wisely. You can picture a child with a once relentless disease who now plays and laughs because a correction in their cells brought a paragraph back to sense. These are not promises for everyone or for every condition, but they are honest directions of travel, built on data, on restraint, and on shared decisions. As we close the book for tonight, hold the idea that life's text is precious, and that editing is at its best when it is quiet, precise, and necessary. Hold the idea that power and responsibility must rise together, that transparency is a companion to progress, and that community consent is part of any step that touches the common future. Hold the idea that science is a craft, not a race, and that careful work with open hands and open ledgers can lighten suffering while honoring what we inherit. Let your breath be the metronome, slow and even, as the workshop dims and the library sleeps. The page stays where it is, the letters rest, and somewhere a small lamp keeps watch, ready to guide the next careful repair when dawn returns.